everyone, here in this lecture we will talk about the structure of placenta. So in this video I will show you how easily you can draw the figure of placenta because it is very important topic for the exam. So firstly I will show you in 3D animation what is the gross anatomy of placenta, that is what are the surfaces, that is the fetal and maternal surfaces, and later on I will draw here the picture of internal structure of the placenta. So you can see and easily draw by your own self. So please watch this video completely and carefully so that you may be able to understand. So here in this 3D animation you can see the term fetus here, and in term it means around 38 to 40 weeks of gestation. This placenta looks circular, that is it is discoid in shape, and the diameter of this is about 15 to 20 centimeter, and it looks spongy because it contains the maternal blood inside the villus system. In center it is more thick in comparison to periphery. So in center it is about 3 centimeter thick, and it has got two surfaces. One toward the fetus is the fetal surface, and the another which faces the maternal uterine wall is the maternal surface, and the peripheral margin which connect these two surfaces, and as you can see here it is attached to the superior part of the body of the uterus, and this weighing about 500 gram. In proportion to the fetal weight it is about 1/6. As the fetus at term is about 3 kg, then it would be 500 gram. So here is the fetal surface, and as it faces toward the fetus, so this surface is more smooth and glistening as this covered by a fetal membrane that is called the amnion. So it is more smooth, and on a mid or in a center the umbilical cord is inserted here, and from the umbilical cord there are various blood vessels radiating from this umbilical cord is easily visible through this amnion. The other surface is the maternal surface, here it is, and this surface is dull red in color as it contains the maternal blood, and this is formed by the decidua basalis, that is the compact and the spongy layer of the functional layer in the endometrium. On its maternal surface there are various lobes or the compartments, and these compartments are called the cotyledons. These are the functional unit of the placenta as I am drawing here. They contain the stem villi within them. So the cotyledons are separated by the decidual septum. We will see later on in the internal structure how this decidual septums are formed. So here in this gross structure of placenta we have seen two surfaces, one is the fetal and the another is the maternal surface, and the peripheral margin which connect the basal plate to the chorionic plate, and the separation of this placenta is going to take place at the term by the spongy layer of this functional layer in the endometrium. So this is how this placenta occupies 30% of the space in the uterine wall. So we have seen the majority of the part of this placenta is formed from the fetus and few part is formed from the mother. Okay, let's see the internal structure of placenta here. So placenta is made up of two plates, that is in inner there is a chorionic plate which is facing toward the fetus, and the outer one is called the basal plate which faces toward the maternal uterine wall. So let's see here how I am drawing the diagram in an easy manner. So there it is the chorionic plate, and in the chorionic plate the innermost structure is the primitive mesenchyme which is formed from the extraembryonic mesoderm. So this is the extraembryonic mesodermal lining, and this extraembryonic mesodermal lining contains the umbilical vessels. These are the umbilical vessels because these are the branches of the umbilical arteries and the vein. So these are the branches of umbilical vessels and the arteries and vein here. So these are lying in the innermost lining of the chorionic plate, and they arises from the umbilical cord, that is from the umbilical artery and the veins. And outer to that is the cytotrophoblast layer. Here it is, and as I am drawing here the tertiary villi, so it is in a branching manner. So these are the tertiary villi. It is the middle part in this placenta. Okay, so here it is the cytotrophoblast. As you know, cytotrophoblast is the mononucleated cells. So I am drawing here in a simple manner so you can draw very easily. And outer to this cytotrophoblast is the syncytiotrophoblast layer. This is the syncytiotrophoblast layer, which is multinucleated. The outer lining of trophoblast, that is syncytiotrophoblast. This blue one. So these syncytiotrophoblast layers are multinucleated because the cell nucleus and cytoplasm become mixed together. There is no exact cell boundaries in between two. So here it is the syncytiotrophoblasts, the outer lining of these villi. So here in this figure you can see what is chorionic plate from here to here. See what is chorionic plate? The innermost lining is the primitive mesenchyme, that is the extraembryonic mesoderm containing the umbilical vessels, and the next is the cytotrophoblast, and inner to that is the syncytiotrophoblast. And this inner, that is primitive mesenchyme, is internally covered by a thin cuboidal epithelial lining which forms the inner fetal membrane. What is this? This is amnion. What it is? The amnion. So here we have drawn the chorionic plate, that is primitive mesenchyme, the innermost, this black one containing the umbilical vessels, the cytoplasm, the black one, and the blue one is the syncytiotrophoblasts, and this is the middle part, that is the villi. Now what is happening? Some of the cytotrophoblastic cells penetrate the syncytiotrophoblastic cells and they arrange transversely like this over the over the syncytiotrophoblastic layer, and forms what it is formed? It forms the cytotrophoblast shell. What it is? Cytotrophoblast shell. So some of these cytotrophoblasts penetrate syncytiotrophoblasts and run transversely and cover this whole villus and forms the shell-like structure which is called the cytotrophoblastic shell. And along with that, the syncytiotrophoblast layer is also there, and at the edges of the placenta they become fuse here and here also. So this is how they lines the intervillous spaces. Now what is this part? This is a basal plate. So what is in the basal plate? Here it is the decidua basalis, very important part. What it is? The decidua basalis. It is the functional layer of endometrial lining. Decidua basalis, functional layer, which is formed from the compact and the spongy layer of the endometrium. So here it is the basalis, and outer to this basal layer is the one thin layer is the stratum basal in endometrium, and outer to that is the muscular layer of the uterine wall is the myometrium. So here it is the myometrium. So in the basal plate, what are the layers from outside to inward? There is the decidua basalis, this part, the decidua basalis, and there is the degenerative layer of syncytiotrophoblast here. Here it is some degenerative cells which is formed from the syncytiotrophoblasts, and the fibrinoid degenerative layer which is forming here is called the Nitabux layer. So what it is? It is the fibrinoid degenerative layer of syncytiotrophoblast which lies in between the cytotrophoblastic shell, as well as the decidua basalis. So in the basal plate, the outer one is the decidua basalis, then comes the Nitabux layer, then comes the cytotrophoblastic shell, here it is, and lastly the syncytiotrophoblast. So we have seen the chorionic plate and the basal plate, and the structure which is lying in between these two plates is called the stem villi, and they develop from primary, then to secondary, and then to tertiary villi. So in tertiary villi, what is happening here? The villus capillary network, starts here, which is very important because these are the structure which help in exchange of nutrient and waste in between the mother as well as the fetus. So here it is the villus capillary network. So here are the spaces in between these villi, and the spaces between these villi is called the intervillous space. And in my previous video I already told you that these spaces are the lacunae, that is the spaces in between the syncytiotrophoblastic cells. So these lacunae merge to form the lacunar network, and later on these become the intervillous spaces. So these are the intervillous spaces, and these syncytotrophoblast cell also erode some of the maternal blood vessels, and as they erode, what is happening? The maternal capillaries, the vessels, that is the spiral arteries as well as the spiral veins, drain into this intervillous spaces. So these are the branches of uterine vessels which is eroded by the syncytiotrophoblast layer, and finally they drain blood into this intervillous spaces, and through the arteries they carries oxygen as well as the nutrient for the fetus to receive by this villus system, and the waste which is coming through these vessels is easily diffused into this intervillous spaces, and then it is drained into this veins. Now what is happening here? This whole villus system is usually arises from the chorionic plate itself. So here in this figure you can see these all villus arises from this chorionic plate, but some of the villi anchored or touches this basal plate. So the villus which touches this basal plate is called the anchoring villi, and some of the villi they remain in this intervillous spaces, they do not reach up till this basal plate. So these are called nutritive villi. So some are the anchoring villi and some of are the nutritive villi. These villi form many branches over here, and as these branch off, they provide larger surface area for the exchange of nutrient and waste in between the mother as well as fetus, and this is about 10 to 14 square meter long areas for the exchange of nutrient. Along with that, this capillary network is also so extensive that it is long up to 50 kilometers. So this is how you can imagine how long structures are coiled up and it is tightly packed inside a woman's uterine wall. Now here some of the decidual tissues goes inside this intervillous spaces and forms the decidual septum. What are they? These are the extensions of the decidua basalis tissues, and these are extending or invading inside this intervillous space from this basal line, but these do not touches this chorionic plate. So what is forming here? Septas are forming here. So these are the septas, that is the decidual septa. So the spaces in between the septum. So here is one septa and the another one is this. So the spaces in between these two septa is the functional unit of the placenta, and that is called the cotyledon or the lobes. So here you can see from here to here, from this septum to this septum, this structure is called the cotyledon. So in the placenta there are 15 to 20 cotyledons or lobes are there, and they are the fissures which make the placenta into compartments, and in each compartment there are 3 to 4 stem villi, but here I have drawn only one. So in each septus, or you can say in each cotyledons, there are 3 to 4 stem villi. So roughly you can say that if there are 20 cotyledons, then there will be 60 stem villi present in the whole placenta. So larger area to exchange of waste as well as the nutrients. So this type of villus system is forming all around the uterus, but it is majorly forming around the superior part of the uterus. So what is happening here? As this fetus grows more and more, this placenta becomes discoid in shape, and here the trophoblastic linings become fused together. Here you can see these become fused together, and the fused trophoblastic lining and the mesenchymal tissues, these are the mesenchymal tissues, and the fused trophoblastic cells forms at the edge of the placenta the chorionic leaf. So what is forming? Chorion leaf. That is the fusion of the trophoblastic layer as well as the mesenchymal tissues. So here also we'll see this is the fusion, and the vessels are also attaches with this lining. Here it is. And another structure that make this placenta to be attached with the fetus is the umbilical cord. So umbilical cord is also lined by the extra embryonic mesodermal tissues, but these become filled up with the mucopolysaccharide or gelatinous fluid, which forms the Wharton's jelly here. And this cord contains the 4 vessels initially, that is the 2 arteries and 1 vein, but later on at the time of 4th month, 1 vein, that is the right vein, become disappear, and the 3 is only remaining, 3 vessels, that is 2 umbilical arteries and 1 umbilical vein. So here the arteries are responsible for exchange of waste material with the mother, whereas the veins carries oxygenated blood. And the length of this umbilical cord varies from 30 to 100 centimeter, but on an average it is considered that it is about 40 to 50 centimeter long. So we have seen the 2 plates, that is, one is the chorionic plate, here it is, I'll just mention here. This plate, that is from the primitive mesenchyme, then the cytotrophoblast and the syncytiotrophoblast forms the chorionic plate, and the another plate toward the mother's uterine wall is the basal plate. What is the basal plate? That is this decidua basalis, then here you can see this decidua basalis, then nita layer of the fibrinoid degenerative layer of the syncytiotrophoblastic, the cytotrophoblastic shell as well as the syncytiotrophoblastic. So this layer is called the basal plate. And the structure which is lying in between these 2 plates is the villi, that is the stem villi, and the spaces in between these villi are called the intervillous space. So the insertion of this umbilical cord is start from the center part of this chorionic plate. So usually it arises from the central, or it is near about the central part of this chorionic plate, and it is covered by a thin layer of amnion here. So by this video I hope you will be able to draw this figure of placenta very easily. So here we have seen the structure of placenta, that is, it is formed from the basal plate as well as the chorionic plate and the villus system in between these 2 plates. Thank you.